All right, let's jump right into this. Today, we're diving into a really fascinating explainer about the ghosts in our genes. We're looking at how our ancestors' past experiences might actually be living inside our biology right now. For decades, we all basically thought our genes were just a static blueprint, carved in stone, unchangeable. But as we're going to see, emerging science is revealing a picture that is way more dynamic than we ever realized.
Imagine for a second that your stress, your specific anxieties, or maybe even your totally irrational fears weren't actually formed by events in your own lifetime. What if you inherited them directly from your parents or your grandparents? I mean, it sounds a bit like science fiction, right? But the research we're unpacking today shows that the profound experiences of our ancestors can actually leave a literal molecular fingerprint on our DNA. To figure out how this works, we first need to understand the difference between genetics and epigenetics. Think of your genetics as a fixed instruction manual. That's the actual sequence of your DNA. Epigenetics, on the other hand, are like a bunch of chemical sticky notes and highlighters shoved into that manual. They tell your body which instructions to read, which to totally ignore, and how loud to turn the volume up on certain genes. So the sequence itself doesn't change, but how it behaves absolutely does. Now here is where it gets really interesting. Environmental factors, things like severe trauma, extreme stress, or starvation, can actually trigger these epigenetic changes. And those sticky notes can be passed down through multiple generations. This is what we call epigenetic inheritance. It means a grandparent's environment can directly influence a grandchild's development without a single mutation in the underlying genetic code. Okay, part 1, mice, memory, and fear. Let's look at how this actually plays out. To test this out, scientists ran an incredible and honestly slightly spooky laboratory experiment. In a groundbreaking study, neurobiologists basically trained male mice to fear the sweet cherry almond scent of a chemical called acetophenone. They just paired the scent with a mild electric shock, and pretty soon, as you'd expect, the mice learned to shudder just from smelling that cherry almond scent alone.
But watch how this cascades. Step 1 is what we just talked about. The original fathers learn the fearful association. But then, in step 2, the offspring of those mice shuddered violently the very first time they were exposed to that same cherry almond scent. They had never been shocked, never even smelt it before. And it doesn't stop there. In step 3, that exact same fearful reaction was passed down to the grandchildren. They were literally born with a biological memory of a threat they had never faced. So what was actually happening inside these mice? Well, the trauma caused literal physical changes. The descendant mice actually grew more scent-detecting neurons, and the fear-processing structures in their brains were enlarged. The researchers traced this back to specific epigenetic marks, altered DNA methylation on the odor receptor gene, and these tiny molecules called small non-coding RNAs in the father's sperm. The fear was transmitted across generations on a cellular level.
Moving on to part 2. The human cost of trauma.
Now, obviously, this phenomenon isn't just limited to lab mice. We have to ask how this applies to humans, especially those who've endured unimaginable historical horrors. Dr. Rachel Yehuda, a neuroscientist who spent decades studying trauma, really captured this beautifully. Her team's work with Holocaust survivors and their children confirmed that human trauma leaves a deep intergenerational biological scar. It fundamentally shapes us on a molecular level. Let's look at the hard data for a second. Researchers focused on the FKBP5 gene, which is directly linked to PTSD and depression. And what they found was fascinating. Holocaust survivors had 10% higher methylation on this gene compared to a control group. But their adult children? They actually had 7.7% lower methylation. They had distinct but completely opposite epigenetic marks. This proves that the intense environment the parents survived chemically altered their children's genetic expression. But, and this is a really crucial point, this data shows us that the children of survivors aren't just simply born with a fixed PTSD-like biology. It's way more nuanced than that. They inherit a highly complex mix of trauma-induced vulnerabilities, sure, but they also inherit traits that promote incredible resilience. Basically, it's an evolutionary adaptation. The biology is trying to prepare the offspring for a world it perceives as dangerous.
All right, part 3, how epigenetics works. Let's get into the mechanics of this.
How does an outside experience literally rewire the brain? Well, it all starts with the HPA axis. This is your body's central neuroendocrine stress response system. When someone experiences severe stress or childhood trauma, it fundamentally disrupts this axis. It basically traps the body in a persistent state of hypervigilance and drastically messes with how your body releases cortisol, the stress hormone. You're either flooded with it constantly or completely depleted. And if we zoom all the way in on the DNA, we see the real mechanics. Remember those sticky notes we talked about earlier? Stress physically alters them. Processes like DNA methylation usually silence certain genes, while DNA demethylation or histone acetylation can turn genes on. So when trauma causes these modifications, especially to things like the glucocorticoid receptor, it permanently dials your body's stress response up or down. And these molecular changes don't just stay microscopic. They scale up and actually alter the physical structure of your brain. Trauma can shrink activity in the prefrontal cortex, the logical, rational, decision-making part of your brain. At the very same time, it traps the amygdala, your fear center, in this constant fight or flight mode. This perfectly explains why trauma survivors can sometimes misinterpret a completely safe situation as a severe immediate threat. Which brings us to part 4, reversing the damage. Now you might be thinking, okay, this is pretty heavy. And it is. But if our biology can be so deeply damaged by a negative environment, what happens if we actively change our environment for the better? This is probably the most hopeful twist in this entire explainer, because it's not a one-way street. Just as severe trauma adds these stressful marks, positive environmental enrichment can actively reverse them. Good experiences literally act as an eraser. A landmark study by Dr. Isabel Mansuhi proved this beautifully. Her team took traumatized mice and, as adults, placed them in low-stress, highly enriched, pleasant environments. The results were absolutely incredible. Not only did the adult mice return to normal behavior, and not only were their DNA methylation patterns completely corrected, but the transmission of those trauma symptoms to their offspring was entirely prevented. No fear passed down. Period. So for us humans, the path forward is actually quite clear. Things like therapeutic interventions, mindfulness, having a strong support system, and, get this, even regular voluntary aerobic exercise act as powerful positive epigenetic triggers. These aren't just feel-good activities. They chemically reduce fear memory and literally rewrite your DNA's sticky notes. You can actively heal your epigenome just by changing your lifestyle and your environment. So I want to leave you with this final thought to chew on. Your genes are not your destiny. They're constantly listening to you, reacting to your environment, and adapting. While the ghosts of the past may influence us, the power to reverse trauma and shape a much healthier, more resilient future, both for yourself and the generations that follow you, is entirely in your hands. What choices will you make today? Thanks so much for joining me for this explainer, and I'll catch you in the next one.